At Metabolics, we're building an AI-powered biosimulation platform to accelerate combinational drug discovery and repurposing. Let's explore its capabilities through a precision oncology use case. The first step involves reconstructing biological networks from publicly available knowledge bases, encompassing many thousand chemical reactions and molecular species. These reconstructions serve as healthy reference templates. Next, we leverage multiomics data to build patient-specific biological networks. For instance, we utilize data from a recent study that analyzed expression data from nearly 1,000 patients across 25 cancer types, using the pan-cancer analysis of whole genomes database. Several methods can then be applied to derive chemical reaction rates. Our patented methodology finally transforms static networks into dynamic models. We developed a self-consistent parametrization strategy using sampling techniques and data from various sources, including metabolomic, kinetic, thermodynamic, and more. Model parameters are rigorously validated by comparing simulated results with available experimental data. What we're after is to observe how cellular phenotypes transition from a healthy reference state to the patient-specific disease state, and vice versa. This is a simplified illustration, but we're not just working with abstract relations. Each edge and node in the graph represents real chemical reactions between molecular species, such as glucose phosphorylation by hexokinase 1, HEX1, illustrated here. Mathematically, we generate a balance equation for each molecular species, as its time derivative, and a rate equation for each chemical reaction, incorporating the data-driven parameters previously mentioned. With our mechanistic models, we can now simulate the temporal dynamics of a cell's molecular state using ordinary differential equation solvers. On the left, you see the rate of a specific enzyme, HEX1, and next to it, several molecular species. This is just a tiny fraction of the entire biological network, which consists of thousands of reactions and molecular species. In steady state, nothing changes. However, by taking an action, such as increasing the rate of HEX1, we can force a shift in a specific reaction. This could represent a drug effect or a gene expression change. Introducing a perturbation, even in just one reaction, triggers a complex response across the entire cellular network. Clearly, predicting the effects of hundreds of differentially expressed genes is far from intuitive. Similarly, we can reverse the action, inhibit the enzyme, and observe the system returning to its original state. With our genome-scale simulations in place, we employ a deep reinforcement learning algorithm to explore the vast combinatorial space of biological networks. On the left, we have an agent that takes actions by altering reaction rates. As seen earlier, each action triggers a complex cellular response, which the agent receives as an observation. Using deep neural networks, the agent learns to map each observation to the next action. We guide this learning process with a reward, or score, based on how effectively the agent's actions shift the disease state toward the healthy reference state. This training process unfolds over multiple episodes. Let's look at an example of training over 10,000 episodes. Initially, the agent knows nothing and takes random actions, resulting in rewards close to zero. Over time, the agent learns which reactions need modulation to maximize the reward. In our cancer example, the agent quickly discovers that targeting multiple reactions gives the highest rewards, indicating that successful strategies involve combinations of targets. Each time the agent achieves a high reward, we can trace the sequence of actions it took, revealing the combination of specific targets that can revert the diseased phenotype. Here's a small excerpt of the results, showcasing a few rows from tables like the one presented. In brief, we obtain target combinations along with their corresponding scores. In this example involving cancer patients, we identified numerous cases where available drugs, whether investigational or already approved, are indicated as antineoplastic agents, which can be useful for drug repurposing programs. As an illustration from our cancer example, the algorithm recommended targeting mitochondrial metabolism and its role in metabolic reprogramming. In this context, the oxidation of glucose-derived pyruvate is replaced by beta-oxidation of fatty acids, whereas TCA cycle intermediates used for biosynthetic processes are replenished through anaplerotic glutaminolysis. The proposed drugs directly engage these pathways and are currently being repurposed for cancer treatment as monotherapies. Notably, our algorithm suggests that these drugs should preferably be used in combination. While this is an average result, our platform enables the computational pipeline to operate at the single patient level, offering significant potential for precision oncology and better patient outcomes.